told me is out of generous. He's very generous to me, and he told you several things about me. Thank him very much for telling about me. Now I will get into the talk. I will share the slides. First, I will try to do that, and then I will start my talk. Let's see. So, are you able to see the screen? Yeah, I am just sharing.
Yeah. It's coming. It's coming up. Yeah.
Okay. Just let me.
Yeah.
You are able to see the slides?
Not yet. You'll
Okay.
have to.
Okay. Now.
Yeah. Now we can see. Yes. Yes. So put it on a full slide mode.
Yeah, that's what I am doing.
Yeah. First thing, you hide your this thing, next to the sharing one.
Okay.
That one, hide that one, because that will keep appearing. Yeah.
Okay. Now let me see.
Yeah.
Are we able to see the screen? I think I'm not yet seeing.
Lovely. Yeah, we can see it. Yeah.
Now it's okay?
Yeah, lovely. Yeah. Again, it went back to put to the full screen.
Okay, so I can continue now? Yeah.
Yeah,
Are you
sure.
able to see this? Okay, thank you very much for the kind introduction, and I welcome all the students to this particular session. We will have some 5-day, 6-day program of this. In the morning session, I will explain you the theory and what we are doing, and in the afternoon session 2 to 4, I will initially brief you the techniques that we are using in proteomics, and subsequently I will show you some demo, some instruments and how we use them. This is generally divided between these two, and at the end of this course or whatever this workshop, I expect at least all the students, those who have attended, should be able to at least start some of these experiments on their own. It is not very difficult, but only thing is you require good instruments and other things. Even without instruments also certain things can be done, so that you will be knowing towards the end of this complete workshop. So today the first talk, I will be focusing on amino acids, peptides and proteins. These are the amino acids are the building blocks of proteins, the smallest building block of a protein. And the structure of an amino acid, which most of you might have studied, this is the structure of a amino acid with alpha carbonatum, which is attached to a carboxylate group and an amino group. These are the two functional groups that an amino acid contains. That means it contains an amino group and an acid group. That somebody is speaking. Can you mute that? And if somebody has any questions during the talk also, you can interrupt me, so that I will be able to explain at that itself or towards the end, depending on your convenience and your interest. Okay, so here this is labeled that this is the Jitraionic structure of the amino acid. This is the alpha amino group, because this is the alpha carbonatum CH, and this is the carboxylate group. The side chain, the structure of the side chain is the one that decides the name of the individual amino acid. So this is the general structure of the amino acids that proteins are composed of, and there are 20 naturally occurring amino acids. All are proteogenic amino acids. Often we also get, instead of a cysteine, we get the selenocysteine. So this is a derivative of cysteine, and some of them are essential amino acids also. As far as the proteomics part is concerned, this is we don't worry, all amino acids are important. We will see those things. And this is the structures of all the amino acids. And you see, you can categorize them into different categories. See, like these two negative amino acids, which are aspartic acid and glutamic acid. They have a side chain of a carboxylic acid group, which carries a negative charge. So these are the negatively charged amino acids. And if you look at these things, histidine, arginine, and lysine, where the side chains are different, epsilon amino group it has here. This is give positive charge for the amino acids. So why these are important is, in subsequent analysis of mass spectrometry, these amino acids plays crucial role on the net charge on the peptide or the net charge on the protein. So these charged amino acids play an important role in the formation of multiple charged residues, multiply charged ions in mass spectrometry. When we go there, I will explain you in more detail. So it is important that these amino acids have a 3-letter code and a single letter code. Mostly, for all practical purposes, we use the single letter code, which I will subsequently show you. For example, this arginine is called as R, histidine is H, lysine is K. So these things you can remember these tables maybe. Did you study these amino acid structures? Students, can anybody can answer? If
Yes,
you
sir.
You have studied these structures. Okay, that's good. So that means you have a background information on this. So therefore, amino acids we have polar and uncharged chains. This is hydrophobic amino acids like alanine, glycine, isoleucine, leucine. So these are all they contain hydrophobic side chains. We also have aromatic amino acids, phenylalanine, tryptophan, tyrosine. These aromatic amino acids will also help us in the estimation of proteins. And for example, if you look at the tryptophan or tyrosine, they have some fluorescent properties also. So for characterizing these proteins, for their biophysical characterizations, these properties are often exploited to study their structure-function relationships. In most of the structure-function relationships, what we focus in proteomics is mainly on the primary structure of the protein. Having seen are these structures of all these amino acids, as I was mentioning to you, this is also an important part that they have single letter code, a triple letter code, and each one, the molecular weight also is written. Here, this first one is the molecular weight of the amino acid, and this is minus H2O, which is generally they present in the peptide or protein chains as called as residues. This is residue weight. This is residue weight, this is the total amino acid weight. So it is important, particularly in mass spectrometry, this mass of these individual residues are very important. Even though it is not important to get by heart all these things, it is important to know the salient features of these things while we interpret the spectral data. And the significant and most important thing in the amino acids, in the first I was showing you that they contain an amino group and a carboxylate group. One is basic and the other one is acidic. These two react together and they form, usually they call an amide bond with the water loss. Water is lost during this and it forms a CONH bond. This is the planar structure in the protein. This CONH bond is found between two amino acids. And this reaction continues between different amino acids, and this is called a dipeptide. And if another amino acid is added here, it's a tripeptide. So likewise, the peptide chain can increase, and there are different ways of synthesizing these peptides, which I did in the lab. Subsequently, after Kannan taught me that sequencing, I also learned peptide synthesis subsequently, and these peptides can be made in the laboratory. And I made a peptide of about 38 amino acids, defensin, one peptide which contains 3 disulfide bridges, very complex synthesis. Such kind of things can be done in the laboratory. What we see in these structures can be made in the lab. That is very interesting. So when we look at the structure of a protein, as I was mentioning to you, this is the dipeptide, tripeptide, and then polypeptide, and these are the proteins. In this particular protein, this is an RNase A, this is the structure, and what we have shown here is a single letter code. See, this is how the protein is made of. This is the amino terminal, this is called amino terminal, and this carboxyl terminal is called the carboxyl terminal residue. So here the amino terminal residue is K, lysine, and subsequently glutamic acid, threonine, alanine. Likewise, we will be able to read the complete sequence in the order. So the order in which these amino acids are present is called the primary structure of a protein. So the identification of the primary structure of a protein is very important. Essentially, what we do in proteomics is identification of the protein. Even though we don't do the primary sequence this way, the primary sequence, how we determine these primary sequence, we use Edman degradation chemistry, and this is how you use the primary structure of a protein. In the Edman degradation chemistry, what happens is phenyl isothiocyanate is the reagent used that reacts specifically with the amino group of the protein. So when it forms an amino group, it becomes a PTC derivative, and this phenyl thiocarbamide derivative is selectively the first amino acid is cleaved under strong acidic conditions. These reactions usually being carried out in gas phase also. So we use it to have automated gas phase protein sequenators. So selectively it removes the first amino acid. For example, in this particular case, if we do the primary structure determination, it relieves the glycine from the peptide chain, and the second residue proline will be new amino terminal is generated. So this derivative, what is generally this phenyl thiocarbamide derivative, is an unstable derivative. It is converted into a stable phenyl thiohydroxide derivative under mild acidic conditions, and then the residue is now fed to the chromatography. There, in the chromatography, you separate all these 20 amino acids. Each amino acid elute at a different retention time, and based on the retention time for the particular residue, we can determine what is the amino acid that is present. Similarly, the chain continues the cyclic reaction. Then PITC reacts with the second residue, and same cycle of reactions follows, and it releases the second residue. Likewise, in the Sedgman degradation chemistry, the first, second, third, fourth, likewise, in a sequential fashion, we release the amino acids from the amino terminal, and we can do sequence towards the carboxyl terminal. So this is how we carry out the primary structure determination of the proteins. And for doing that, usually what people also do, they contain disulfide bridges. For example, here we can see only one cysteine. If it has one more cysteine, it can react with that cysteine and form a disulfide bridge. In such kind of cases, we reduce those disulfide bonds and make also iodoacetylide treatment, and then we linearize the protein and then use it for the sequencing. And that derivative, whatever the derivative that we use, whether it is iodoacetamide or iodoacetic acid, depending on the retention time, they can be subsequently identified on the protein sequenator. And if you have to do this particular procedure, you require to purify the protein almost to 99% pure. If there is any contaminant protein or any other contaminant, then you get at the N-terminal itself, you get two residues. If you have two proteins, you get two residues. Then it is very difficult for you to obtain the sequence. So for obtaining the primary structure of a protein, one of the essential criteria is to purify the protein to homogeneity. And we have to use several chromatographic methods, ammonium sulfate precipitations. Likewise, we have to have a scheme of things for protein purification. That itself is a big area. You have to isolate the protein from the mixture and purify further and purify to homogeneity, so that you will have 99 point or above 99% pure. Then only you can carry out the sequence. But if you take a cell, biological cell, it will have lots of proteins. Can anybody guess? For example, if I give you an E. coli cell, bacterial cell is a prokaryote, and I say it contains around 3,500 genes, can you tell me how many proteins it can synthesize? Any students, no problem, you can answer. Don't
So
worry,
it's
wrong.
more than 35,000 genes.
35,000, 3,500, 3,500 genes it contains. How many proteins can it synthesize theoretically? Yeah, answer, no problem. Even if it is wrong, it doesn't matter.
So probably it will be more than 35,000.
Okay, any other answer? Anybody else? Okay. Actually, what happens in prokaryotes and eukaryotes, there is a difference in the transcription and translation. In prokaryotes, usually, one gene, it can produce one protein. Whereas in the case of eukaryotic cells, you have RNA processing. So DNA, from DNA, RNA is produced, that is messenger RNA, and this messenger RNA undergoes maturation due to introns and exons. So therefore, due to these permeabilities, one gene in a eukaryote can give more than one protein. Whereas in the case of prokaryotes, it gives one gene, one protein. So if I say E. coli, something unicellular bacterium at prokaryote, 3,500, theoretically, it will produce 3,500 genes. But in general, when we use bacteria and when we are using, actually, 1/3 of the genes are generally expressed in prokaryotes. 1/3 of the genes are expressed. So if you have 3,500, maybe 1,250 or 1,100, approximately that many number of proteins are synthesized in the cell. Okay, why I am telling you, for a protein sequencing, if we have to identify the protein sequence using Edman degradation chemistry, we will require to purify the protein. The project itself will take around 6 to 8 months. Protein purification is not an easy job. You have to purify this, and then only you can carry out the structural identification of the protein. Whereas, if you look at any biological systems, they synthesize large number of proteins. For example, the case in a bacteria, they can have around 3 to 4,000 genes usually do they have, and they contains, even if you say 1/3, 1,500, 1,000 to 1,500 or around 2,000 depending on the bacteria that we have, that many number of proteins they produce. This is one complexity. And another thing, after the protein is synthesized in a biological cell, it undergoes post-translational modifications. In order to carry out specific reactions or specific functions, these proteins undergo post-translational modification, like phosphorylations, like acetylations, geranylations, palmitoylations, several kinds of modifications they undergo. So these modifications also needs to be identified. And when we do protein sequencing using Edman degradation chemistry, the elution profile of these derivative amino acids or these modified amino acids is rather complex, and sometimes we may not be able to fully detect them. So these are the some limitations that are present in the primary structure determination of those things. And these are some aspects. For example, now if we go to eukaryotic cells, for example, if I give a human cell from a particular tissue, and if I say human cell contains around 23,000 genes approximately, how many proteins it can produce? Now I told you the difference between eukaryotes and prokaryotes. Eukaryotes will have 4 times or 5 times. We do not know, depending on the RNA processing and subsequently protein modifications, isoforms. So how many proteins it can produce if it has 22,000 or 23,000 genes? Anybody guess? Students, you can mention. There is nothing wrong.
Around 20,000 to 21,000 proteins will be produced.
How many? 20?
20,000 to 21,000.
No, I told you it contains 23,000 genes approximately. And I said that the gene undergoes RNA, it produces DNA, produces RNA, and that undergoes further maturation with introns and exons leaving, and then further formation, and the final transcript is transcribed. So one mRNA can give you more than one protein, isn't it? It can give 4 proteins or 5 proteins, yes or no? Because this RNA has to mature. The mRNA forms from DNA,
Yes,
and that contains introns and exons. Introns are the ones which remain, exons are the ones which leaves from the RNA, and the final mature RNA is the one that gives you the transcript, and that gives you the translate into protein.
Then
Okay.
sir, it will be around 1,20,000.
Yeah, see, so now you are able to at least guess something. So one RNA is not one protein in eukaryotes, whereas in prokaryotes that is true. So these are the number of proteins, and in addition, their isoforms, their post-translational modifications. See, the complexity is more. So if we really has to understand the biology of a single cell or a biology of a system, you require to understand all the proteins that are present in them. Because we know how to do the complete genome sequence is now known. In the human genome, complete genome is known. For example, if a bacteria is given, we can sequence the bacterial genome maybe in a matter of a few days. Nowadays, we have the sequencers that work with that powerful methods and most accurate, and we can do the sequencing. So this I am telling you, the complexity that is involved in the identification of proteins, the numbers that is happening. This is point 1. Point, I think this you have appreciated. So a bacteria can produce around, if 3,000, 4,000 genes can produce around 1,000 to 1,500 proteins, of course, plus their post-translational modification. That is the number that we have to handle. And when we have to identify human systems or these eukaryotic systems, they give you, if you know n number of genes, it can give you 3n or 4n number of proteins. and also their post-translational modifications. So the numbers are much more. One hand, the number of proteins that requires to be handled. And now another point that I will highlight here. For example, if you take a bacteria or a human cell, if it is producing, let us say, bacterial cell producing 1500 proteins. Now, all the proteins will not be of the same concentration. Some proteins will be produced, synthesized at a higher concentration, and some proteins are synthesized at a lower concentration. So, therefore, there is a range of concentration of proteins that are there to be identified. So, within a cell, if you take, so what are the how do you measure the concentrations? Either if you can weigh, you can say milligrams, micrograms, that is the scale, or in molarities. For example, when we say picomoles, femtomoles, nanomoles, micromoles, millimoles, so these are the scales. So in a particular system, you can have a dynamic range of protein concentrations, starting from a femtomole protein to a nanomole or a micromole in a bacteria. So there are two complexities that are present. Number one, the number of proteins. Number two, the dynamic range, the concentration range that is present, and post-translation mode. So one is the numbers you have to handle. Second is the concentration that you have to handle. When we use this Edman degradation chemistry, we use it to do it at a nanomole concentrations. If we require a 1 or 2 nanomoles or 5 nanomoles is the protein usually, or 1 nanomole. That is the range that we use it to do. So if you have less concentration, you will not be able to do from N-terminal to carboxyl terminal. You require concentration. And also sequencing from N-terminal to carboxyl terminal is also not a single step. Even though the reaction can carry out, depending on the size of the protein that you have, it takes time. Now, in such case, what do? You first do N-terminal amino acid of the proteins to whatever extent that you do, then subject it to a proteolytic digestion. When I am now focusing on the single protein again. Now I told you the complexity between eukaryotic and prokaryotic system, both in terms of the expression of proteins and also the dynamic range of concentrations. Now that is one problem that we need to handle. Now another problem that I am focusing on in how to identify the primary structure. Primary structure, N-terminal to carboxyl terminal, if you put it on an automated sequencer, one residue 45 minutes to 1 hour we use it to take. So in a day, we use it to identify around 24-25 residues per day. And if you have a 100 residue or 200 residue protein, all residues will not be coming in one go, because this is called a repetitive yield. From first residue to second residue to third residue, when you go out, the yield will be reducing. That is one problem. Second problem, the reactivity of PITC is not identical to all the residues. For example, there is a proline, you may have to do a double coupling. That means you have to increase the temperature. So likewise, you have to program the sequencer in such a way that you will be able to get it. And you will not be knowing when you are using an unknown sample where these residues are. So you have to do these experiments multiple times. Then you will be able to get a confident data. And complete protein sequencing in one go is not possible. Then we need to subject it to a proteolytic cleavage. For example, using something, an enzyme like trypsin. Trypsin usually cleaves the proteins at the carboxyl terminal of lysine and arginine when there is no proline adjacent to them. So this is generally, theoretically, that is what it says. So if you take a protein, if you take a protein, and for example, I say lysine plus arginine, I have 22 residues. How many peptides can I generate from a protein? Are you hearing? I have a protein which contains lysine plus arginines. Assuming that there is no proline closer to that, number is 22. How many peptides can I generate from the protein? I will repeat again. I have a protein and I am subjecting it to the tryptic digestion. And in tryptic digestion, trypsin hydrolyzes the carboxyl terminal of lysine and arginine, carboxyl terminals. So it cuts the protein at that rate and it generates the peptide. And the protein that I am using contains 22 lysines plus arginines. So how many peptides can I generate? Okay, I will give you another simple question. I have a peptide with me which has one lysine in between. Okay? If I digest with the trypsin, how many peptides does it give? In the middle of the peptide, I have one lysine. Are you hearing the talk? Or I hope you are all. Hello?
Yes, sir, we are hearing.
Did you understand the question? I
So
have a
we'll
big.
get two lysine, I think. Two
No, what?
lysine. If we cleave a peptide with two lysine molecules, then we will get two protein molecules of lysine.
Okay, you hear to me again. Again, I will tell you. I have a polypeptide with me. It contains only one lysine in the middle, and I subject it to a tryptic digestion. And trypsin cleaves at lysine. Okay, how many peptides can this polypeptide will become? One polypeptide. I subject it to tryptic digestion, which contains only one lysine. So, how many peptides can I generate?
So three, like two peptides and one lysine molecule.
Yes, yes. Two peptides is the right answer because it clears it carboxyl terminal of the lysine. So therefore, it gives me two peptides. For example, if I have one lysine and one arginine differentially spaced between for a 15 to 20 residues, how many peptides do I get? I have a polypeptide which contains one lysine and one arginine. Now, how many peptides do I get?
So three.
Correct. So that means if I have one lysine, it will give two peptides. If I have lysine and arginine, it will give three peptides. If I have 22 lysine and arginines in a protein, how many peptides it gives?
So 23.
Absolutely. This is the point that I want to get from you. So therefore, one protein if you take, it will give me so many peptides. Okay. So now you take those peptides, purify those individual peptides on HPLC, and again carry out the Edman degradation. Okay. Now the entire protein, you will be getting various peptide sequences from various regions. Now you have to subject the protein with another proteolytic enzyme, okay, which have another specificity. For example, I can say, okay, you need not have to use only enzymes. You can also use chemical reagents also like cyanogen bromide. Cyanogen bromide cleaves at methionine. Similarly, it gives you, depending on the number of methionines that it contains, n plus 1 number of peptides, and those peptides also have to be sequenced. Now you have sequence of peptides which are generated from tryptic digestion, and now you sequence of peptides from the same protein which is cleaved at methionine, another set. Now N-terminal region, you know, N-terminal to 1 to 50 or 1 to 40, you have the N-terminal sequencing. And now you see whether there are any lysines or trypsins from N-terminal in between. And from there onwards, you have to look at the peptides that contains either from trypsin or from methionine and overlap. Did you follow what I said? For construction of the complete sequence of the protein, you have to prepare the set of proteins from tryptic digestion, and you have to prepare the set of peptides from methionine or whatever another digestion that you have done. And now you know the N-terminal sequencing, and from N-terminal sequencing you will be able to do, because of the automated sequencing, 50 residues or 40 residues. Within that 50, you may have lysine, arginines, or methionines. And from there onwards, what is the sequence that you have got? That you get when you do these peptide sequences. So you have to keep on overlapping these regions. Could you get the logic?
Yes, sir.
Huh? First, you have to do the N-terminal sequencing of the protein. You get as much information as possible. Then generate tryptic peptides, generate sequence of all those peptides, and put a list with you. Then you generate with another proteolytic enzyme like otherwise cyanogen bromide with methionine. Generate peptides. Methionine actually gives larger fragments, so it is easy to do larger segment sequence. And then generate those sequences. Now, if you from N-terminal you start and overlapping from both these sequences that you have, then you will be able to construct the sequence of the protein from carboxyl terminal to from N-terminal to carboxyl terminal. Could you get this point? This used to take a lot of time in the laboratory. Earlier, people used to do the sequencing with this particular procedure. This is how people used to do. Of course, subsequently, other methods also developed for doing carboxyl terminal sequencing also, which is not very successful, but you can use carboxy peptidases, which releases the amino acids from carboxyl terminal, and you can identify those residues also, and conclusively identify the primary structure of the protein using this method. But this is a laborious, time consuming, and requires huge amounts of proteins. Okay, this is what is the procedure that was in the identification of the proteins. This I have already told. And also in this procedure, what we also do, we usually hydrolyze the protein with 6N-HCl, which Kannan was mentioning that we did extensively for several proteins. So this 6N-HCl cleaves all the peptide bonds. So you get complete protein amines. All peptide bonds are broken. So you essentially get amino acid composition of the protein. Okay, this amino acid composition is determined by 6N-HCl, and primary structure is determined by Edman degradation. Combining these two, we can correlate these two results, and finally we can validate the structure of the protein also. So this is how people use it to do the primary structure determination. Now if you see, for example, if you take two different proteins, you get amino acid composition like this. How many individual amino acids are present? This is grams per 100 grams of protein. You can actually estimate the protein by this method, and also you can see the amino acid composition. Now, so far, what I have told you is now this I have already mentioned. And okay, so this is how people use it to do the degradation, Edman degradation chemistry, and then identify the protein structure. And this I already told you, the disulfide bridges also broken before doing that with dithiotritol and iodoacetamide, and we linearize the protein for doing the N-terminal sequencing. These are the reactions how they use it to carry out, and this we use it to do in the lab. And subsequently, this is what the procedure that I have told you. This is the automated sequencer that we have, and these are only the reactions which I was talking to you. Those are the reactions that they form and the different derivatives, and you identify those things. But this also has a limitation, this procedure. For example, if you have a pyroglutamic acid at the N-terminal, or the N-terminal is modified by acetylation or some other modification, then you will not be able to apply this method for the primary structure determination. Then you may have to modify those things and then carry out the reaction. So now you are able to now appreciate the complexity that is involved. This is for one protein. What we are doing, all this procedure is for one protein. And if we have to do for multiple proteins, how long will it take? For one protein itself, you will take 6 months or 1 year to do all these procedure, Edman degradation, reduction of disulfide bridges, and then establishing the disulfide bridges. There are several methods. See, these are various enzymes like trypsin, chymotrypsin, pepsin, cyanogen bromide, BNPSS ketol. These two are chemicals. CNBR specifically acts on methionine, and BNPSS ketol acts on tryptophan. So we can generate peptides. So that means different kinds of peptides can be generated. Now, if you want to sequence the protein using these particular procedures, actually it will take us for us sometimes 1 year or even more time or 6 months. However fast you do, because you have to purify the peptides, individual peptides have to be sequenced, assemble them together. This all will take time. Now, all of you, can you understand how to do the sequencing of a protein? Or anybody has any questions so far? Please feel free to ask the question, because once you are clear, then I will move further. I will go to the next step. Hello?
Sir,
Any question?
yes, sir. So you mentioned something about repetitive yield and problem in complete protein sequencing. Can you elaborate on that?
Right. So, for example, when you take a pure protein and put it for the Edman degradation on the automated sequencer, what happens is first residue, when it couples almost 100%, and first residue completely is released from the protein chain, and the second residue is generated, and in the second residue, depending on the nature of the residue, the coupling reaction itself may be 98%, or maybe 99%, or maybe 96%. So what happens? you are able to do 96% of the protein only is N-terminal reacting, and the remaining 4%, is remaining with the second residue, first residue only. It is not reacting. It is remaining as it is. So what happens when you do a third residue? The 4% or 3% or even less than that will still be retaining, and you will be getting third residue. That is one. That is the background. And the yield from 98% to 96%. So 96% to 94%. Likewise, the yield of the residues also keep falling, because the efficiency of the reaction from residue to residue it will vary. And when you keep these cycles in repetition, during repetition also, the efficiency of the reaction keep reducing. Of course, it is more than 95% or more than 98%. Very efficient automatic protein sequencer. In spite of that, there is a loss of this one. So with these very high efficient and high sensitive instruments, we will be able to do around 50 to 75 residues. Very, very highly efficient reaction. Okay, so did you catch what is the repetitive yield from residue to residue? The yield falling down from first residue to 10th residue, 10th residue to 20th residue, keep on residue falling. Then, since the sensitivity, you will not be able to see the signals also after 50 residues, because yield is fallen down. Because of the gradual falling down of the yield of this reaction, you will not be able to see the after 50 or 60 residues. That depends on the nature of the primary structure and also the efficiency of the reaction, these two coupled together. Did you catch this point? Did you understand now?
Yes, sir. Thank you.
Okay. Okay. Any more questions? Okay. If you don't have any more questions, then this is how we do the sequencing from N-terminal to sequencing, and by using different endopeptidases and exopeptidases, combination of these things, both from carboxyl terminal, you get some information. Using carboxypeptidases, amino acid terminal, this is a PITC reaction. So using this combination of procedures, we will be able to ultimately identify the complete primary structure of the protein. Okay. So this I will give you a list of these proteases that people do use and their source. And these are all can be used for cleaving the peptides, for using proteolytic digestion. You can use all these enzymes, a wide variety of enzymes are available. And the tryptic digestion, which I was speaking to you, this is how it does the tryptic digestion, and it cannot cleave when there is Kp or Rp. And this tryptic digestion is an important issue, because this we will take it forward when we go for proteomics also. And actually, this tryptic digestion also, the specificity of the trypsin also has been extensively studied. In some cases, what happens is there is a non-specific cleavage when we use this trypsin, and also based on the protein conformation, some sites are exposed where you can digest that particular residue. When these lysine arginine residues are buried inside the protein tertiary or quaternary structures, then accessibility of the trypsin may not be there, and there will be missed cleavages. In the peptides which have missed cleavages, you still see lysine or arginine residues. In non-specific cuts, other than lysine and arginine also, sometimes they do generate. These are some limitations of the tryptic digestion. And then people subsequently identify the disulfide bridges. This I have told you. This is transcription and translation. And one gene, one protein in prokaryotes, whereas one gene giving multiple proteins in eukaryotes. This is the background information that we have. In a cartoonic picture, we showed that you have a biological cell where there is a nucleus, and from there you have chromosomes and they transcribe. And proteins, when they function, proteins can function individually like your enzymes and other things, or they function as complexes also. So some proteins will form complexes for carrying out some biological functions. So the technology that we are thinking about, the technology that we want, should be able to identify the complexes. So if we use only advanced degradation chemistry, identifying this large number of proteins in thousands and thousands will take infinite time. So it is very difficult to do sequence and identify the primary structure function. It takes huge time. And another thing, post-translational modifications have to be determined separately by using separate chemistries. And protein complexes and concentration ranges. So now we have, these are the, as the complexity is increasing, as we go, when we want to understand the biology of a cell from the proteins point of view, and to understand them, to identify all of them, the complexity is increasing, and we require to have some technology which answers all these questions. That means the technology that we want to use now, let me see the complexity which you have already addressed. That, for example, if human genome has 20,000 or even more number of proteins, and the protein expression, this is another complexity. Now we have only seen so far that the number of proteins, the dynamic range, and the chemical modifications and complexes. So these are now, are you able to follow how many points where the complexity is increasing from protein identification? Number of proteins is one aspect. Post-translational modifications is another aspect. Protein complexes is another aspect. And the dynamic range from femtograms to nanogram present in the same place. So the technology that we want to identify these things should accommodate, should be able to accommodate all these features and should give us the result. In addition to this, in addition to these common features, when we study eukaryotic cells, these cells often, the protein expression in these cells, which we have not considered so far, is depends on various factors. That is environmental factors, the pH of the surrounding, the drug administration, stress conditions, aging, and disease. For example, if we take human body, we can say approximately around 200 cell types, let us say, a few cell types, if you omit, around 200 cell types. Each cell will have the same DNA, but the proteins that they express is different. Depending on the organ that it forms, the kind of proteins that it expresses is different. The post-translational modification is different. And the quantity of protein, for example, present in different cells, different organs. In one organ, the cells makeup of the proteins may be of a different concentration. In some other cells, it may be of a different concentration. And some of them it may not even express. So these are the various things that we should understand. And not only that, the protein expression is depending on aging. As the aging takes place from young to adult to old, these changes will be coming, including post-translational modifications. That is one thing. Second thing, environmental factors. For example, if we stand in sunlight for some time, the different kind of protein expression. If you go and stand in a cold room in your lab, you can get a different set of proteins. So therefore, the expression of proteins in a cell is depending on the environment. And for example, if a person is diseased, you do drug administration. Then the protein expression where you target this drug, that particular organ may synthesize different proteins. So under disease conditions and under normal conditions, the protein expressions changes. And this is what is exploited in determining the biomarkers, protein biomarkers. Okay, that means the expression of proteins under different conditions will help us to identify and understand the processes in detail. So if you have a protein, a normal cell and a cancerous cell, because normal cell, unregulated growth, it forms, it turns out into a cancerous cell. So the protein expression is different between these two. So if you can identify the proteins from the normal cell and also from the cancerous cell, you will be able to identify selectively or specifically expressing proteins in the cancerous cell, and you will identify what are those proteins. So all these features can be accommodated in the proteomics technologies. Okay, so these are the various complexities that we need to understand, and the technology that we are going to talk about, or the technology that we are going to develop, should be able to address all these questions. And now time factor is another thing that we are talking about. Protein sequencing, when we say, is taking 6 months to 1 year. But now those time periods are no more valid because you have to do so many hundreds and thousands of proteins. So time should reduce, sensitivity should increase, complexity should be able to handle. So using all these features, we should be able to identify all the proteins. That is what the proteomics technology offers. Proteomics technology offers to identify all the proteins at a given time of a particular cell, at a particular situation, including its post-translational modifications, and if it has any complexities and its isoforms, you can identify all of them, even if they are of dynamic range from picogram to nanogram concentration. A different wide spectrum of concentration range we will be able to handle. So this proteomics technology will help us to identify all these complex features that we require to understand from biological cells. And that is very important for the technology. And for example, if you even proteome has not yet been completely done. But human genome sequence has been done. We know the complete human genome sequence. And human proteome, I will tell you, the proteome shows copy number of proteins per cell. You can even calculate that. How many number of individual proteins present in a particular cell also can be calculated. And most abundant proteins are identified. And now I will tell you the scale, because I am telling you the complex scale. You also should appreciate what is this scale. 1 mole is 1000 millimolar. Because now why I am telling you this, I am now slowly taking you towards proteomics. And we use, these are the concentrations. What concentrations we have? Very low concentrations are the ones which we use at that time, we should know. So 1 millimolar is 1000 micromolar. And 1 micromolar is 1000 nanomole. 1 nanomole is 1000 picomoles. 1 picomole is 1000 femtomoles. 1 femtomole is 1000 attomoles. And 1 attomole is 1000 zeptomoles. The proteomics technology that we are using are sensitive enough to detect at the zeptomole concentration. You can imagine whether it is contamination, whether it is protein, very, very difficult. We are reaching to such a level of concentrations. Very difficult even to imagine. You can't even see any material at that concentration. Whether it is protein or any other molecule, at this concentration, very difficult to visualize these molecules. But you get the complete structural information at those concentrations. So therefore, it is very, very important to appreciate that we will be able to identify large number of proteins are in proteomics, all proteins that are present in a given cell at a given time. Why a given cell and time? Because time is dynamic. If you take bacteria from different growth stages, the proteins varies. So when you are doing proteomics of bacteria, you have to fix some parameters. At what stage you are doing the proteomics, that has to be fixed. Then only you do. Otherwise, you get different results. So it is another complexity that we should be able to analyze. So the technology that we are talking about, large number of proteins and individual specific, different concentration ranges, cell type related, age related, disease related, isoforms, and including post-translational modifications, and also the protein complexes. All these things can be detected using proteomics technology. And what it is, just let us wait and we will get into that. And at this stage, I will stop. And if you have any questions, feel free to ask me. Then we'll move forward. Yeah, now that the lecture has ended.
Yeah. So, Jagannadham, beautiful presentation.
Thank you.
And I really enjoyed listening to myself. I could see Jagannadham speaking. It was very nice. You spoke very well, very good presentation. Children, are you able to correlate with the 23rd June lecture you heard when you joined this program? Hello?
Yes, sir.
Now, what Jagannadham took you slowly through all the things. He will take you through in the next for another 4 days. He will teach you what we were talking in that 2-hour lecture.
Yeah.
You know, so that is what you are going to see, and you will be able to follow what. So that lecture, please revise, and now you will become clarity. Number 2, I suggest that you take a molecule like say ubiquitin, for example. Okay, it's a very small polypeptide, or what some people call it, protein. See, with trypsin, he gave you a list of enzymes. So go and look at the sequence and try to predict how many tryptic peptides will be produced, how many type of chymotryptic peptide will be produced, how many cyanogen bromide peptides will be produced. Try with various enzymes which he showed you, and then make a report on that, because that will tell you have understood how it cuts. Now remember one thing which is very important: trypsin is an endopeptidase. Okay, so it is a endo mean, if suppose a lysine at the amino terminus, it will not cut. Okay, he kept on saying lysine is in the middle. Okay, he did not say it is in the corner, because if it is in the corner, the trypsin cannot cut that. So you have to be very, very careful that suppose the amino terminus is lysine, it will not cut at lysine after that. Okay, so just remember this. So just look at it, and I suggest you take a molecule like ubiquitin, go and look for the sequence, because it's a simple molecule to handle, and try to predict how many peptides will be produced. Okay. Then today, I'm sure he will take you through the rest of them in the afternoon. He'll show you. So thank you so much, Jagannadham. We'll catch up at 2 o'clock. Am I right?
Here I will make a small announcement for afternoon class.
Yes.
So in this class ends. In the afternoon, what I will do, I will take you to the electrophoresis, which is one of the components of proteomics, one of the important components of proteomics. And I will tell you how to run the gels briefly for a 15 to 20 minutes. I will brief you on various technologies that we use. Subsequently, I will show you the apparatus, and I will show how do we do the analysis. Okay, I will show you the instruments, and I will take you to various instruments and show you how to do that. Is like a semi demo. We will not be able to do the complete practical work, but I will tell you how to do all those things in the afternoon session. So every day in the morning we will have a lecture like this, and in the afternoon session we will have the training kind. Okay, so training means some, as now Kannan is mentioning to you, you can take any protein and digest those things, and in one class I will tell you how to do all those things in an automated fashion. That also is included in the program. So we'll do that. So if you don't have any questions, we'll stop at this, and I see you at 2 p.m. Okay?
Yeah. Yeah. Thank you, Jagannadham. See you at 2. Yeah.
Thank you.
Thank you, sir.